Hello, this is talk number 7 in the series from Human Pathology by Robbins and Cotran, 9th edition. Today we'll be discussing autoimmune diseases. We will be discussing immune rejection and immune deficiency situations. We'll first start with autoimmune diseases. This requires 3 things: an immune reaction that's specific for self-antigen or self-tissue. The reaction is of primary pathological significance, and if no other cause exists, it can be systemic or organ-specific. Here are some examples of what we're referring to. There's autoimmune hemolytic anemia, systemic lupus erythematosus, autoimmune thrombocytopenia, myasthenia gravis, Graves' disease, Goodpasture syndrome, type 1 diabetes mellitus, rheumatoid arthritis, multiple sclerosis, scleroderma, Sjogren's syndrome, and postulated immune diseases, Crohn's disease, ulcerative colitis, which are flip sides of similar processes, primary biliary cirrhosis, polyarteritis nodosa, and autoimmune chronic active hepatitis. So there are organ-specific versions here, systemic versions here, T-cell immune mediation, autoimmune probable antibody mediated here. Immunologic tolerance. There's a lack of inflammatory response to self-antigens. Central tolerance has self-reactive T and B lymphocytes. These are killed or removed should they recognize self-antigens. This is negative selection, or it's an example of receptor editing in B cells. Peripheral tolerance is where lymphocytes are rendered unresponsive. This is called anergy. T-cell activation requires antigen interacting with self-major histocompatibility molecules and a second signal to stimulate a response. This does not occur in anergy. The process also occurs in peripheral B cells. Regulatory T-cell suppressors are usually CD4 T-cells, which co-express CD25. These help in acceptance of the fetus during gestation. Removal of self-antigen T-cells is a form of apoptosis. Autoimmune mechanisms require susceptibility genes and environmental triggers, such as infections or tissue damage. Susceptibility genes are multigenic. There are HLA alleles associated in some diseases. Others have non-MHC associations with polymorphisms. Other genes are lost. We call these knockout, and they lead to autoimmune disease. And there's a role of infections which cause activation of factors and cells which break down anergy. Here's some features of autoimmune disease. There's genetic susceptibility genes, failure of self-tolerance, and self-reactive lymphocytes. There are also environmental stimuli, infections, tissue injury, inflammation, which act on tissue. There is activation of tissue APCs, an influx of self-reactive lymphocytes into tissue, and activation of self-reactive lymphocytes, and then finally tissue injury. The model autoimmune disease is systemic autoimmune disease. Systemic lupus erythematosus is a systemic multi-organ disease with immune complex deposition and antibody binding. The hallmark is the formation of autoantibodies, particularly anti-nuclear antibodies in SLE and related diseases. An antigen-antibody pattern varies by disease. It can determine with indirect fluorescence, genetic predisposition, multifactorial immune complex type 3 site hypersensitivity, can have thromboses from antiphospholipid antibody. This is the schematic again. Susceptibility genes with T and B cells specific for self-nuclear antigens. Also external triggers such as UV light, radiation, which is a form of hypersensitivity, apoptosis of cells, which is defective clearance of apoptotic bodies, increased burden of nuclear antigens. You get anti-nuclear antibody and antigen-antibody complexes. And then after that, you have interaction with B cells and dendritic cells with endocytosis of antigen-antibody complexes and TLR engagements by nuclear antigens, TLR stimulation B cells and DCA, and stimulation B cells and T cells by IFN. So these are type 1 interferons acting on this when you get persistent high-level anti-nuclear IgG antibody production. Tissue transplant rejection is triggered by HLA allele differences. There's allograft where the same species is involved in transfer. Xenograft is where a different species is involved. There's allo recognition. The direct pathway has recipient T cells which recognize donor dendritic cell antigens, and then an indirect pathway where recipient T cells recognize secondary signals from antigen-presenting cells. There are two types of rejection. There's a T cell-mediated rejection, acute and chronic rejection. Acute rejection has CD8 cytotoxic lymphocytes directly killing graft cells, which occurs within a month. Chronic rejection, cytokines release, and they induce vascular endothelium and smooth muscle. Antibody-mediated rejection, there's hyperacute rejection, where preformed anti-donor antibodies are present in circulation. These are seen in prior transplant cases which share HLA alleles. Acute antibody-mediated rejection, autoantibodies are formed against the donor, class 1 and class 2 HLA antigens. Chronic antibody-mediated rejection has an insidious clinical course. Amelioration of rejection is HLA matching for kidney. Other factors are used for other organs. Immunosuppressive drugs inhibit cytokines. Complications of immunosuppression include infections, some neoplasms, graft versus host disease, where the donor is immunocompetent and the recipient is non-immunocompetent, so the donor tissue attacks the recipient tissue. Immunodeficiencies are primary and secondary types. The primary immunodeficiency diseases are genetic. They affect different cells and cell functions. There's leukocyte adhesion, phagolysosome function, microbiocidal activity, and signaling. You also see complement deficiencies and defects. There's a lymphocyte maturation defects, a spectrum of humoral and or cellular defects, a combined and a sex-linked type. You can have lack of B-cell maturation, lymphocyte activation, and lack of antibody production. The disease immunosuppression model to cover is AIDS. This is a global pandemic, 40 million prevalence, 35 million total deaths to date. There's sexual and bloodborne transmission, and particular interest is infection in newborns. HIV is a retrovirus. There's HIV-1 in USA, Europe, and Central Africa, HIV-2 in Western Africa and India. The structure is a lipid envelope with glycoproteins. The inner core has 2 RNA copies, capsid proteins, and viral enzymes. This is a view of it. There's the GP41, GP120, the capsid layer, lipid bilayer here, integrase involved in the unwinding of the RNA strand. The RNA strand is here. There's the second one. Reverse transcriptase produces a DNA complementary template from the RNA primer. And this is a remarkably rapidly mutating virus, and there's no good, obvious locus from which to direct a vaccine against. There's considerable variation in the genome. It targets the immune system and the central nervous system. CD4 cells are infected and lost. It binds to CD4 T cells and cytokine receptors. Conformational changes allow viral entry into those cells. The reverse transcriptase activity precedes DNA integration. RNA transcription products buds out of the cell, and it leads to cell death. Here's the virus entry, the CD4 chemokine receptor. This is a virus with GP41 and GP120. There's binding to the CD4 conformational change. GP140 and CD4 bind to CCR5. There's GP41 membrane penetration and membrane fusion, and the RNA genome is extruded into the cytoplasm. Reverse transcriptase produces a DNA template. This gets integrated into the host cell genome, and there's an HIV DNA provirus which is formed, and this undergoes transcription to RNA, and the RNA is involved with the synthesis of HIV proteins. They assemble the virion core. Finally, you get budding and release, and a new HIV virion. And this is an accelerated, amplified process, and this leads to the death of the cell in question. So there's viral entry, viral replication, viral release, and this is mediated with cytokines and cytokine receptors, and this activates the cell with transcription of the HIV genome. These infect non-activated T cells, but it is latent until the T cells are activated. HIV is cytotoxic to the CD4 helper T cells. There are minor functional effects on other cells and organs. HIV infects macrophages and dendritic cells. It acts as a reservoir of viral particles and infection portals. B cells activate and proliferate in response. In the central nervous system, HIV affects macrophages and microglia. Neural damage is of indeterminate cause. In the disease course, memory CD4 T cells are infected on mucosal tissue. Humoral and cell-mediated responses occur. There is seroconversion in 3 to 7 weeks. A mild, self-limited flu-like illness which is called acute retroviral syndrome. This occurs 3 to 6 weeks after infection, and it resolves in 2 to 4 weeks. Viral load is a marker of the disease course. A constant set point is reached following treatment. This rises with clinical worsening, and the CD4 T cell count is an additional index. HIV replicates in lymph nodes and spleen during the latency period. AIDS is acquired immunodeficiency syndrome, and this is the final phase. It consists of fever, weight loss, diarrhea, secondary infections, neoplasm, and neurologic disease. These are all hallmarks of AIDS. The therapy is a combination directed against reverse transcriptase, protease, and integrase. These are AIDS-defining opportunistic infections. You could get protozoal and helminth infections, cryptosporidiosis, which I had a post-mortem with that is the cause of death in HIV. Pneumocystis carinii pneumonia, which has particular histologic appearance of the microscope. Toxoplasmosis manifests either as pneumonia or more commonly central nervous system. Fungal infections, which run the gamut, candidiasis, cryptococcus, coccidiomycosis, and histoplasmosis. Bacterial infections, you get mycobacteria, usually atypical, such as Mycobacterium avium intracellulare, which is either disseminated or extrapulmonary, Mycobacterium tuberculosis, which is either pulmonary or extrapulmonary. Narcoidiosis is another one, salmonella. Viral infection, San virus is common. Herpes simplex is common. Varicella, progressive multifocal leukoencephalopathy. Kaposi sarcoma, primary lymphoma, and invasive cancer of uterine, cervix, or other possibilities which can and do occur. Kaposi's with a particular a spatial representation which distinguishes it from the historical type of Kaposi's, which is a different disease in terms of etiology, different distribution, different demographics. Amyloidosis is a fibrillar protein deposition. This causes tissue damage and functional compromise, an associated group of inherited and inflammatory conditions. These are associated with multiple myeloma. It's a deposit in that disease. Alzheimer's disease, which is a sine qua non of detection under histologic means, and an acute phase response, also seen in heredofamilial amyloidosis. And that's it. Thank you very much.